So in the course of this talk today, we're going to be looking at the way in which plants deal with environmental stress. And I'm going to be looking at it on two different levels. So the first level is about what you might call short-term, about how plants adapt themselves more or less immediately to the environments that they find themselves in, particularly with respect to finding themselves in stressful environments. And the second, probably the last third of the talk, is more about the long-term effects of what, if you are a plant growing in a stressful environment, does to you, not in terms of you yourself, but in terms of subsequent generations. So it's a kind of long-term evolutionary consequence of plants living in stressful environments. So the sort of stress that I'll be talking about all of the time is salinity stress. So plants are affected by the levels of salt in the soil, and soil salinity affects around 6% of the land area of the world to varying degrees of intensity from high, shown in red here, downwards. And that salinity affects plant growth, very much affects crop plant growth. So current estimates are that soil salinity affects around 20% of global farmland, so it has negative impacts on the growth of crops grown on that land. And it's a problem that actually is getting worse, because not only is there certain soils naturally contain high levels of salt, but increasingly, as we need to feed ourselves as a human population, we need to use irrigation in order to grow our crops. And irrigating land increases the salinity of that land, because usually we use river water, and that river water contains within it solutes. That water evaporates, and as a result of that, the solute content of the soil gradually increases. So this is a problem that's increasing, and one that we need to deal with. One of the reasons why we need to deal with it is that most crops are very sensitive to salinity. We know that very broadly you can divide plant species into halophytes, so those are plants that are very well able to deal with salt in the soil, and glycophytes, so plants like these crops here, which are very poorly adapted to dealing with salt in the soil. And if you look at this graph here, this is a halophyte plant here, so what you've got here is its kind of tolerance, if you like, to salt on the x-axis, and on the y-axis you've got increasing concentrations of salt. And you can see this plant is relatively tolerant. Whereas most of our crop plants, so here we have wheat, durum wheat, bread wheat, barley, rice, they're relatively sensitive to salt. And also you can see here that the plant Arabidopsis thaliana, which is a plant that I'll be talking about mostly today, is also very relatively sensitive to salt. Its sensitivity to salt is pretty high, and so therefore in some ways it can be taken as a model or as a parallel, if you like, to some of our most important crop plant species. So we tend to think of soil salinity as a modern problem, but actually it's not. It's been a problem that's of historical proportion. So this is an example of a civilization, the Sumerian civilization on the rivers Ur and Tigris, that existed around 3,200 BC, and that civilization declined, and archaeologists believe that its decline was at least correlated with increase in soil salinity that occurred here as a result of irrigation from those rivers, salt going onto the land, gradually meaning that the crops that they used to grow, they couldn't grow there any longer. So you can think of that, if you like, as a kind of archaeological or historical model for what may be facing us as a global civilization. Okay, so as I said, we're going to divide this into short-term physiological adaptation, by which I mean mechanisms for tolerance in some ways or other, and later on I'm going to be talking about evolutionary aspects. So, as I already said, I'm a Arabidopsis geneticist. A lot of what I do involves using the genetics of Arabidopsis to try to understand how plants grow or work, or to understand their biology. And so there have been many genetic mutant screens for Arabidopsis mutants which are altered in the way that they deal with salt that they find in their environment. And many of these screens have taken place using Arabidopsis grown on Petri plates containing agar, containing various concentrations of salt in the agar. And as a result of that, many very important discoveries have been made. So we know, for example, about this system called SOS1, SOS2, and SOS3. These are proteins that we now are genetically identified that exist in the cells of plants that control the way in which plants deal with finding themselves in a high salt environment. And in many cases, like SOS1, for example, and also another protein called HKT1, these proteins are transporters. They affect the way in which the plant transports the ion, the sodium ion, from outside into the cells of its plant body. And so these have actually been very important, not just from our understanding of fundamental biology, but they're actually been important practically as well. So this gene HKT1, in the last year or two, Rana Munz and Mark Tester in the US, in Australia, have developed increased salt-resistant durum wheat, so that's pasta wheat, as a result of using variant forms of this HKT1 transporter. So these experiments, originally conducted in the model plants like Arabidopsis and so on, have fed through already to practical developments in improving the salt tolerance of wheat. So I want to emphasize that these previous experiments were very important. But we came into this area thinking that there were some deficiencies in the way in which these original experiments were done. So we thought that maybe there were things that would be missed by these particular mutant screens. And in many ways, our ideas were encapsulated not in print by this review again by Rana Munz and Mark Tester. Many of the experiments, so the ones I was describing just now in Arabidopsis, were conducted on plants grown on agar, in Petri plates. And as a result of that, because the plate is enclosed, there's no transpiration going on. Okay? And of course, transpiration is massively important to plants growing in the real world, out in fields. And we already knew that transpiring conditions have a major influence on sodium transport and on tolerance to the sodium that plants find in the soil. And so we set out about exploiting this idea. So just to remind you, here's a cartoon of a plant. It's got its roots in the soil. Those roots absorb minerals, including salt. Salt then travels up in the xylem sap, up the xylem vessels in the vascular system of the plant, via transpiration, where transpiration or evaporation of water in the leaves causes the transpirational flow, which moves the salt and the minerals in the xylem sap from the roots up to the shoot of the plant. So we kind of exploited this by doing the mutant screens in a different way. And so what we did, and this is a picture now of our glasshouse in Oxford, is took Arabidopsis plants, grew them on soil in a normal environment where transpiration is occurring, and watered them with salt solution. So this is a so-called M2 population. So this is a mutagenized population of Arabidopsis. And what we're going to look for now is the very rare mutant examples which differ in the way that they respond to the salt that you've put in the soil. So there's so many plants here, thousands of plants growing here, you can't actually see the individual mutants because they're relatively infrequent. But here now are two, you can see in close up. And so the first two-thirds of the talk this morning, I'm going to be focusing on these two mutants. So this one that we call soil salinity sensitive, or initially called it that, and this one that we initially called soil salinity tolerant. And what you can see here is that this sensitive mutant, compared with the plants growing around it, it's a little bit pale green compared with the others around it, and so it's more sensitive to the salt that we've put in the soil. Perhaps rather more obviously, this one, tolerant mutant, that's still alive, whereas all of these other non-mutant plants around it have been killed by the salt that we've put on the soil. And so next 20 minutes or so, I'm going to be talking about these two mutants. I'm going to be explaining to you what they reveal about how the plant deals with salt in its soil, and also showing you how actually they relate to each other. So the mechanism that these two reveals, they both affect the same mechanism. So let's deal first with soil salinity sensitive 1, or SSS1. So as I've said, it's sensitive to soil salinity. So this is a control along here. Here's the mutant grown in normal soil, and here it is 10 days after salt treatment. And now you can see very clearly that that mutant is yellowing, much paler than is the progenitor shown above. And just to go back to the point I'm trying to make here, which is that this is a class of mutant which wouldn't have been identified in the previous mutant screens that were done in Petri plates. What we can now do is take that mutant and grow it under those Petri plate conditions. And here we have now the progenitor, mutant, progenitor, mutant, progenitor here, mutant here, in gradually increasing concentrations of salt. And what you see here is that in the Petri plate conditions, this mutant has there's no difference between it and the progenitor in Petri plates, but as I showed you previously, there's a profound difference between it and the progenitor when it's grown in the transpiration active conditions of normal growth on soil. So because we work with Arabidopsis, and because when I moved to Oxford, my lab got very quickly set up to be able to do whole genome screening or whole genome sequencing, it's actually very easy and very quick for us now to be able to identify the gene that's affected in any new mutant. I'll show you a little bit more about how we use whole genome sequencing later on in the talk. But simply what you do is take the whole genome sequence of your progenitor and the whole genome sequence of your new mutant, and you compare the two and you look for mutations. Very few mutations that arise. And in this case, in our mutant, SSS1, we found that there was a mutation that had caused a kind of very small inversion of a bit of DNA, and that inversion had interrupted this gene here that's called, given that number there on the annotated Arabidopsis genome sequence. So that gave us a very strong clue that our gene, the gene that's affected in this SSS1 mutant, is actually this gene here. And because the Arabidopsis genome is very well sequenced, we know about all of the genes in there already. This was a gene, it turned out, one was already known about, a gene called ATRBOHF. Now, you may say to me, well, okay, you found this mutation, doesn't mean that that mutation is the cause of your mutant phenotype. And so in order to do that, now that we think it's ATRBOHF, the gene that's affected, you can order up from the Arabidopsis stock center new alleles, different plants which carry mutations in that gene. So this is an example of one here. So this is a tDNA, so-called tDNA insertion into ATRBOHF, compared with Columbia, its progenitor. And again, you can see 7 days following salt treatment, it's pale in the same way that our original SSS1 mutant was pale. And indeed, if you make a cross between the two of them, here's the F1 plant, you can see that that plant is pale on salt. So that's proving to you now, it's taking you through the whole process, that you find a mutant, you make a DNA sequence of that mutant, you identify a candidate gene that's affected in that mutant, and then you do the final genetics just to prove that you really have nailed it, that it really is that gene or mutation in that gene that's the cause of your phenotype. So you may be saying to me, okay, so then what is this gene ATRBOHF? What does it do? So ATRBOHF is a gene which encodes an enzyme, an NADPH oxidase. And these enzymes have actually become very famous in plant biology, actually in biology in general, in recent decades, because they catalyze the production of a chemical species called reactive oxygen species. So what the enzyme does is it takes electrons off NADPH, feeds them across a membrane, and oxidizes the oxygen molecule to create superoxide. And as a result of that, a whole variety of so-called reactive oxygen species are then derived from this superoxide. And superoxide molecules are highly reactive chemically. We've known about them in plant biology for a very long time because they're often produced as a result of stress. They're a stress product, superoxide or reactive oxygen species. Most famously, perhaps, they're the product of when plants are under high light stress. So there's got so much energy coming into them that they can't capture it all in photosynthesis. That energy often gets dissipated in the form of reactive oxygen species, which then go on to be actually very damaging. These things are indeed very damaging to proteins and to DNA. They're mutagenic. So they're things that the plant has to be very careful of. And so for a long time they've been known about as stress products in plants. More recently it's become clear that plants actually uses them as sources of information. So that ROS is itself a signalling molecule, that the plant uses it in order to detect whether or not it's in a stressful environment, and can actually use the production of ROS molecules to regulate the expression of genes and to regulate various activities. And so that's these kind of developing ideas are covered quite nicely in this review that I've shown up here. We now know that actually reactive oxygen species are responsible for signaling in a wide variety of different aspects of plant biology. So this is just a picture taken from that review, and I'll just highlight a couple. So we know, for example, that ROS can do systemic signaling, so it moves around the plant and triggers signaling in a different part of the plant from where it originates, or can do that. It's involved in abiotic stress, as I'll be showing you in a bit more detail in a minute. And my colleague Liam Dolan, also at Oxford, is famous for having shown that ROS is involved in controlling root hair formation and elongation. So multiple roles as a signalling molecule, this ROS. And A2RBOHF is one of a whole series of genes in the Arabidopsis genome that encode for these so-called respiratory burst oxidases. So you see a whole series of genes here, all the way from A through to J or H, I can't remember. And you might say to me, well, okay, so the Arabidopsis plant has got all of these enzymes. Is there any specificity with respect to the particular one that we've already identified to do with this salt response? And so you can answer that question by getting, from the stock collections or from elsewhere, mutants that individually lack one or other of these enzymes that are encoded by these different genes. And so this is a panel showing these a whole set of these various different mutants. And we've done now the soil salinity experiment that I talked about at the beginning. And if you look at all of these, here's the control. Here's lots of different knockouts of all these different enzymes. It's only A2RBOHF that has this phenotype. So you can see it there. And you might say to me, well, this plant's got it too. And it has indeed, but that's actually double mutant between A2RBOHF and A2RBOHD. So you need to be lacking A2RBOHF specifically in order to have this soil salinity sensitive phenotype. So we then went on and looked at this in a little bit more detail. And A2RBOHF, first of all, what we did was made fusions between the promoter of the A2RBOHF gene and GUS, glucuronidase, so that you can stain for the GUS activity and use that as a reporter of the activity of the A2RBOHF gene. And what this shows, this picture here, is a longitudinal section of a root, and this is a cross-section. And in red, we've artificially highlighted the endodermal cells of the root, which encase the central stele, the vascular tissue of the root. And if you look at that, you'll see that we have GUS staining in the root that's restricted to within the vascular bundle or the vascular tissue of the root. It's not in the cortex, not in the epidermis of the root, but within the endodermis, in the vascular stele, that's where you get the stain of GUS. And that stain is increased in the presence of salt. So what that's telling you is that A2RBOHF is expressed in the root vasculature, and that that expression is increased by salt. So what does it do in there? You might imagine it makes ROS, that's its job. and you can stain for ROS by using a stain called nitroblue tetrazolium, which stains purple in the presence of ROS. And here you can see now wild type, so normal roots being stained with nitroblue tetrazolium. And if you do that in the absence of salt, you can see a ROS signal, the purple, in the vascular tissue of the root. If you treat that wild type plant with salt, you get an increase in the amount of staining. So salt increases the amount of ROS within the root vasculature. Now, what happens if you do that in a mutant, in our mutant now that lacks A2RBOHF? Well, first of all, you see much reduced ROS signal in the control, but most significantly, you see that you now have much reduced ROS induction by salt. So here's a control. Salt induces ROS in the root vasculature. In the mutant lacking the A2RBOHF enzyme, you don't get an increase in that ROS. And does that have an effect on the levels of salt? So you can actually image salt within the roots of plants using a dye called CoRO-Na. And here we have our wild type plant. And if you look here, this is now a kind of cross-section of the root, if you like. And these are epidermal cells here. And as you see, you give those cells salt, and what you get a lot of salt in the epidermis, you get a lot less inside the plant. Conversely, if you look at our A2RBOHF mutant, you still see lots of salt in the epidermis. Now you also see a bit of salt staining here. That corresponds to the vasculature. So what that's telling you, or indicating, is that in this mutant, so the mutant that can't make ROS in response to salt, you now got more salt in the vasculature than you have in the wild type plant. And you can take this a stage further. You can actually harvest, so this is a way of actually harvesting the xylem sap of the shoots of plants. So these are hydroponically grown Arabidopsis plants, and if you put a little bit of tubing on the stem of the plant, you can collect here the exudate from this thrown out by root pressure from this hydroponically grown plant, and you can chemically assay now that exudate, which is largely xylem sap. So it's a way of measuring xylem sap. And if you do that, here's an experiment where we're doing it hours after salt treatment. Orange here is the wild type plant, so the normal plant, and you can see that gradually the levels of sodium in the xylem sap increases. In your mutant that's lacking the A2RBOHF enzyme, you get a much greater increase in salt. So lack of A2RBOHF reduces the level of ROS in the root vasculature, and that, we believe, in turn, increases the level of salt that gets in or is present in the xylem sap. And that xylem sap now is what gets kind of shot up by the transpiration stream into the shoots of plants. So you might then say, well, is that then reflected in the level of salt that's actually found in the shoots of those plants? And so here now is that measurement. So here's again, what the control is orange, the mutant is in green. And you can see that as you go over time, days after salt treatment, the salt level in our mutant plants lacking the A2RBOHF enzyme is much higher than in the wild type plants. So let's just kind of reflect a bit on this and put it all together. So what we're saying is that salt present in the soil is absorbed by the root and eventually enters the vascular tissue of the root, and then by the transpiration stream travels up the root into the shoots of the plant. And what we're saying is that salinity is somehow perceived, that activates this gene A2RBOHF. The A2RBOHF catalyzes the production of ROS, and that ROS then basically regulates the amount of salt that gets shot up into the rest of the plant by the transpiration stream. So ROS regulates sodium homeostasis and tolerance. And so in our wild type situation, salt taken up by the root travels up the xylem vessels in the transpiration stream to the shoot. In the mutant, salt is taken up, and because there's no ROS in this mutant, so in the wild type, remember, it's making ROS because it's got this active gene enzyme, makes ROS, that reduces the amount of salt in the xylem sap. Here you've got no A2RBOHF produced ROS, as a result of that, more salt in the xylem sap, more salt gets transferred up to the shoot of the plant. And that's why those plants are sensitive. That salt, increased salt, poisons them, makes them yellow. As you remember, the original mutant was yellows compared to the green of the wild type. So that's how this mechanism works, and that sort of explains the phenotype of this first mutant that we identified. And that's all, for those of you who are interested, that's all described in this paper that we published in 2012. So, now we're going to move on to the second mutant that I showed you, and you remember now this is the kind of mirror image, the opposite of the first mutant. This one, unlike the first mutant, this one is tolerant of salt. So here we see our progenitor, and here's the mutant, and you can, it's actually not very clear in this picture. I hope you remember the previous one where this mutant actually is profoundly tolerant of salt in the soil. And as again we did with the previous mutant, we need to know, would we have recovered this mutant if we'd done the screen in Petri plates rather than on soil? And so that's this experiment here. And here's our progenitor, grown in increasing concentrations of salt. And here's the mutant, again, increasing concentrations of salt on a Petri plate. And here again, no difference in phenotype. So what that's telling you is, again, that the phenotype of this mutant that is salt tolerant is dependent on it growing in soil conditions and likely dependent on the activity of the transpiration stream. So once again, whole genome sequencing, very quickly we're able to work out what gene is affected in this mutant. I won't take you through the whole process again because it's just the same as before. But basically, it shows us that this gene that we originally called SST1 is actually a novel allele of a gene that was again already known in Arabidopsis, a gene called ETO. So ETO is a gene where if you lose the function of this gene, your plant makes more ethylene. So ethylene, a very important plant hormone signaling molecule. And basically, as I say, I won't take you through all the genetics here, but essentially ETO, our salt-tolerant mutant, is a mutant, as an ETO mutant, a mutant of the ETO gene. And again, you remember now that the phenotype of ETO is the opposite of the phenotype of the ATORBUHF loss of function mutant. And so does it then have opposite effects on the levels of salt in xylem sap and in the shoot of the plant? And if you just focus on the green and the grey here, so green is the control and grey is the ETO mutant. And here we have days following salt treatment. And this is, again, measure of xylem sap. And what you see now here is that as you increase as the days go by, the salt in the xylem sap of the wild type is appreciably higher than is that of the ETO1 mutant. So that again is indeed the opposite of what ATORBOHF mutant did, in which that mutant, the levels of salt in the xylem sap were higher than the wild type. And so then you can ask the question, so does that xylem sap salt translate into levels of salt in the shoot? And the answer, again, is that it does. So once again, focus on the green and the grey. Green is the wild type, grey is the ETO mutant. This is days following salt treatment. And you see the green wild type plants have much more salt in their shoots than does the ETO mutant. So this ETO gene is affecting the accumulation of salt in the shoot of plants in the opposite way to the way in which the ATORBOHF mutant did. And these mutant plants are tolerant of salt in the soil because they don't transfer as much salt from the soil up to their shoots as the wild type plant does. So you can ask the question now, well, do these two mechanisms relate to one another? Does what ETO does relate to what ATORBOHF does? And I'm a geneticist, so the way I answer this question is to make a double mutant between the two. So the question you might ask is, say, okay, we know that ATORBOHF reduces the level of salt in the xylem sap. We know that ETO increases it. Maybe ETO increase is dependent on the function of the normal ATORBOHF enzyme. And if it is, then then the effect of ETO will be reduced. That tolerance will be abrogated, if you like, if you knock out the A2RBOHF gene. And so that's the experiment that we've got going here. So we have a progenitor plant, a mutant carrying the original A2RBOHF gene, a mutant carrying the ETO gene, and now the double knockout mutant. And so the key question is, what is the phenotype of this double mutant? Does it look like A2RBOHF, or does it look like ETO? And here it is, and you can see it looks like A2RBOHF. So what that's telling you is that loss of function for A2RBOHF suppresses the phenotype, the salt tolerance phenotype of ETO. It makes it salt sensitive in the same way that A2RBOHF is on its own. And once again, you can do the measurements of salt in xylem sap and in shoot. So in green here we have the in orange here we have the wild type, the progenitor control, certain level of salt in the shoot. In the A2RBOHF mutant, as I've already told you, more salt in the shoot. In the ETO mutant, less salt in the shoot, but in the double mutant, the one shown in just here, that one has a level of salt that's very similar to the A2RBOHF mutant. So what that's telling you is that ETO confers its salt tolerance by affecting the activity of the A2RBOHF gene, or it suggests that. And indeed, that is the case. So here we have examples of the levels of transcripts of the ETO gene in the presence of salt and in control conditions. And I've got my finger over it, that's why it's not working. I was about to say the laser printer isn't working. But, and what you see here is that in the presence of salt, you get increase in the relative transcript levels of A2RBOHF, so it increases its level in normal conditions, and in the presence of the ETO mutant, it increases it even higher. And that's similar sort of results are sort of shown here, but I'm going to skip that just in the interest of time. So in a sense, this is really the key experiment to all of this. So here we have, so this is now where we're actually looking at ROS production in these different mutant lines. So here's the control, grown in the absence of salt, the progenitor line. If you take that progenitor with the ETO mutant in it, you have a little bit more ROS within the vascular tissue of the plant. In A2RBOHF mutant, as I've already shown you, you don't get that ROS produced, and that ROS is not produced also in the double mutant. Here in the presence of salt, salt increases the level of ROS. It's very high in the ETO mutant, and it's abolished in those lines that carry A2RBOHF. So that then enables us to put together a kind of picture of all of this. So salinity, likely, or I mean it's already well established in the literature that soil salinity increases the levels of ethylene in plants. That ethylene stimulates the activity of A2RBOHF. RTRBOHF increases the levels of ROS, and ROS then regulates sodium homeostasis and tolerance, homeostasis of salt within the xylem sap, plant tolerance by regulating the amount of salt that gets transferred up to the shoots of plants in that xylem sap. So I think it's worth emphasizing at this point that this is an entirely new mechanism now. I mean, this is not a mechanism that has not been previously described. I think it has profound relevance to plants growing in agricultural conditions, so plants growing in soil as opposed to plants growing in Petri plates. It's very likely that other salt tolerance pathways like the SOS1, SOS2 and so on, things that we already talked about, interact with this as well. And just to sort of emphasize the importance of this thing, this is now plants grown on soil where we're actually comparing plants mutated in the system that we've uncovered, so this A2RBOHF mechanism, with the more widely known SOS1 and SOS2 mechanisms. And you can see these plants are all of a similar level of sensitivity to the salt in the soil. So I think what I'm trying to do here is make a pitch for this mechanism as being as important as some of the other, perhaps more widely known, previously better understood mechanisms of salt tolerance in plants. And I think it's now that we've kind of worked out the fundamental biology of it, I think it's now ripe for exploiting by breeders and others to try to make improved crop tolerance for a whole variety of crop species. So the ethylene stuff that I've just been talking about is described in this paper that came out in September of last year. So finally, you might say to me, and it kind of relates to what I've just been saying about crops and its relevance, okay, this is great, you've worked all this out, but it's all in Arabidopsis. Does this really have any meaning for plants that perhaps we care about more agriculturally? And my answer to that is, I think, probably. And the reason for that is that A2RBOHF likely confers a mechanism for salt tolerance that's evolutionarily conserved. So it's not just a property of Arabidopsis, it's a property of other plant species too. And the reason for me saying that is shown on this slide here. It's not proof, but I think it's nevertheless sufficiently good evidence to make it important that we continue to investigate this. So this is wheat. And these are wheat plants treated with a chemical called DPI. So DPI is a pharmacological inhibitor of those respiratory burst oxidase enzymes that I've been talking about. So the enzymes of which A2RBOHF is one. And so wheat carries an A2RBOHF type enzyme. And here is a control where we've treated control plants, treated control plants with DPI, and now plants treated with salt, and now plants treated with salt and DPI in combination. And these were plants grown hydroponically. And what you can see here is that these wheat plants treated with DPI, which is, if you like, a pharmacological imitator of the Arabidopsis A2RBOHF mutant, when you give them salt, the leaves go pale, in the same way that the Arabidopsis mutant leaves went pale. And you can actually measure, again, the levels of salt in the shoots. So without DPI, in the presence of salt, you get this level of salt in the wheat plant shoots. With DPI, you get a much higher level of salt. So it's all suggestive of the fact that a similar mechanism to what we've uncovered in Arabidopsis is regulating the levels of salt in transpiring wheat plants. And so for that reason, I think my answer to the question, were you to ask it about how relevant this is to crop plants, is that there's great potential for this to be relevant and to be useful in a practical sense in the future. So, okay, a final couple of slides on this topic, and then I'm going to briefly move on to the evolutionary topic. So, what we've identified here is a mechanism that controls the way in which particular solutes, in this case sodium, get into the xylem sap, or at least how they accumulate within the xylem sap. And you might then ask the question, well, is it possible that this mechanism actually regulates other solutes? And of course, solutes getting into xylem sap is an enormously important thing, partly because of the interest in plant nutrition. All of the important ionic nutrients like nitrate and phosphate and so on eventually get into the shoot via the xylem sap. So how they get into the xylem sap is a very important question. So we're kind of broadening our interests now beyond salt itself and trying to understand to what extent this mechanism that we've uncovered affects other ions getting into the xylem sap. And one thing that we've looked at, what I'm showing you here, is heavy metals. And this is a picture of, this is preliminary unpublished data, but this is cadmium. And basically, you can show that this same gene, A2RBOHF, affects the cadmium sensitivity. So cadmium is a toxic heavy metal. A2RBOHF mutant is more sensitive to cadmium than is its wild-type control. And it is so because if you measure the shoot content of cadmium on plants grown on medium containing cadmium, there is more cadmium in the shoots of the A2RBOHF mutant than there is in that of the wild type. So that is suggesting that this same mechanism that's regulating salt getting into the xylem sap is also affecting the levels of cadmium in the xylem sap, and it may be it's affecting many other things as well, and that's something that we're kind of looking into at the moment. So, just to wrap up this bit, what's the future? Well, I mean, there are many, I think, aspects to this that are interesting. I think I'm particularly interested in this idea that what we're actually uncovering now is important from the point of view of the relations between plant and salt. But what was particularly interesting to me now is that I think we're getting into actually understanding how the plant regulates homeostatically regulates its solute content of its xylem sap in general. And I think that's a hugely important and interesting topic, and that's an area where I kind of want us to get into in the future. Another important area is the whole question that I'm sure is going through your minds is how does ROS regulate xylem sap solute content? That's a kind of big gaping question that's been left throughout my talk. I don't know the answer to this. There are many, many areas of plant biology that are regulated by ROS. I think it's fair to say that in no case do we really understand how ROS is regulating the things that it regulates. And so this question, I've written it here about xylem sap content, but it actually applies to all of the other phenomena that we know that ROS regulates. We really don't understand what it does and how it's sensed in particular. And so that's a big question, and that's one that I'd like us to approach. And finally, I think, as I've mentioned, I think there's general applicability to agriculture, to sustainable intensification in terms of salt tolerance breeding, heavy metal tolerance breeding, nutrient use efficiency, and so on and so on. And I think the list goes on. So that's the kind of first two-thirds of the talk. I now briefly want to talk about the kind of evolutionary stuff, which really is a bit of a change of gear. As I say, it's more looking at what happens long-term to lineages of plants that are grown in stressful environments as opposed to short-term adaptation. And so this, of course, is a picture of Charles Darwin taken towards the end of his life in his house at Down House, quite close to here, in fact, in Kent. And I've never been, but one day I'll go. Anyway, of course, you all know that amongst his many great contributions to biology, the very famous book, The Origin of Species. And The Origin of Species talks about how, provides an explanation for the diversity of the natural world. And there are kind of two planks to the argument that it proposes. One is that biological variation arises, and the second part is that then natural selection works on that variation, and you all know about how selection works and evolution is driven. And I think it's fair to say that in that book, and in my view ever since, we've known a huge amount, or we've come to know a huge amount about selection through the work of population biologists and ecologists and so on. But we actually know very little about the first bit, which is about how variation arises in organisms in the first place. And indeed, Darwin himself was very vague about it in his book. In chapter 5, he says something like, variation however it arises. I just basically don't know. And I think to a certain extent this remains true today. So kinds of questions that I'm interested in are to what extent does environmental stress of the kinds that I've just been talking about, so salt stress, affect mutation? So does it affect base substitutions, transitions, transversions, insertions, deletions of DNA and so on? And the second question is to what extent does it affect epimutation? So change in, I'm particularly going to focus here on change in cytosine methylation status. And I just want to emphasize here, what I'm interested in here is stuff that is heritable. I mean, we all already know that stress affects the degree to which genes are methylated in a kind of immediate response sense. What I'm interested in is what change is heritable. That to me is the important question. So we've had now for a number of years set up in my lab experiments, mutation accumulation lineage experiments. So what we have are plants grown, again Arabidopsis, grown in controlled conditions and grown in saline soil conditions. And what we do is just grow them generation by generation. So grow them, take seed, plant them, do the thing again, do it generation after generation over time. And we're actually up to, it just shows generation 9 here. We're actually up to generation 15 at the moment with these experiments. And we have other experiments in which we're doing other sources of stress. So temperature stress, for example, is an important experiment that I won't be talking about today. And so really what we're asking here, at least initially from the point of the mutations, which we'll deal with first, is what kind of mutations accumulate in these lineages? Obviously, as you're going along, mutations will arise. They don't arise very frequently, but they do arise, and gradually they accumulate over time in this lineage as you're going along. And so what kind of mutations, what's the rate of mutations, and does it differ from the control populations and the experimental populations? And so we can detect mutations, and this is why Arabidopsis is the plant to use for this, again by doing whole genome sequencing. So one of the things that I did when I moved from John Innes to Oxford was set up a collaboration with a human Wellcome Trust Centre for Human Genetics in Oxford, and develop the computational ability within my group for us to be able to take the Arabidopsis genome sequence of a progenitor plant and of a next generation plant, and very rapidly, within the course of a few days, use computational algorithms to pinpoint the very infrequent mutations, mutational differences that occur generation by generation. And so this is just a snapshot of one way in which we do it. This is for those of you interested, this is a program called Integrated Genome Viewer. It's developed in the Broad Institute in the US, and we actually customized it a bit to be able to use it specifically with Arabidopsis. And what you see here is this is the Arabidopsis reference genome. So this is just a small, very small chunk of the genome. You can imagine the whole genome could be theoretically spread out like this. You can just see the letters of the genome there. And this is one of our mutation accumulation lineages. And the grey bars here are individual reads from the next generation sequencing machine. And you can see here that there's a difference there between the reference, the original genome, and the mutation accumulation lineage genome here. And I can't actually quite see what that is, but I think it's a nucleotide substitution mutation there. And here, perhaps more easy to see, this is a deletion mutation that you can see. So you can go through the whole genome and spot the mutations, pick them out, identify them, and categorize them, if you like. And so that's what we've been doing. And just to summarize very briefly for you the kind of results that we're getting from this. Basically growing Arabidopsis plants on salt in the kind of levels of salt that I've shown you, which is enough to stress the plants, but obviously not enough to kill them, actually roughly doubles the mutation rate. So this figure here, we had 52 mutations in total in our control populations, and we have 102 in the stressed population. So you have a doubling of the mutation rate in these stressed plants. And it doesn't just increase the rate, it also increases the spectrum, the type of mutations that you get. So for reasons I won't go into at the moment, we could talk about this later, but in this group here, most of these mutations, and this is known or been already published by others, are of a transition type. So these are, most of these are transitions. Transitions, especially GC to AT transitions, predominate in populations of plants grown in non-stress conditions. However, when we grow them in stressful conditions, now actually the transversion rate increases. And in fact, most of this increase here is due to increase in transversions and to indels. So these are little insertions or deletions of DNA sequence that also occur much more frequently in stressed plants. So, so the answer then to what we're showing is that growing these plants, these lineages in a stressful environment increases the mutation rate, and it also changes the spectrum. You get different types of mutations in plants grown in stress conditions. And I think this is important. I think it's important because it means when we're growing crops, we're increasing the mutational load in stressful environments. And I think it's also important because plants, when they're growing in nature, are not growing in controlled environment conditions. I mean, most plants growing in nature are in what is largely a stressful environment. And so I think that estimates of mutation rate that have been developed according to organisms grown in relatively benign laboratory conditions may actually not be telling us about the types of mutations that are the rates and the types of mutations that are accumulating in plants growing in nature, and I think that matters. So I think because of time, forgive me, I'm going to skip the epimutation bit because otherwise I will just go on for ages. So we'll just skip all this and come to the conclusions, which is that so growth on saline soil increases the pace of genome evolution. It increases the frequency of mutation accumulation, in particular by increasing transversion frequency. I didn't show you, but it increases the frequency of methylcytosine epimutations. So why does it matter? Well, different mutational spectra theoretically can have different effects on protein evolution. Theoretical biologists tell us that transversions are in many ways less conservative than transitions, and so therefore they will affect proteins more than transitions do. So that's why that matters. And finally, I think this whole fact that mutation rate is higher within stress populations suggests that we need to rethink the way in which mutations are arising in nature and the way that we think about that. So that's kind of all I had time to say. I wanted to say more, but I think we better cut it short. I'd like to thank people from my group. So Kai-Fu Zhang, fantastic postdoc from China, who's actually just returned to China. He's now has a faculty position at the Chinese Agricultural University in Beijing. Zhang Xiaogan was a postdoc jointly between me and my friend and collaborator Richard Mott from the Wellcome Trust Centre for Human Genetics, who also did a lot with respect to the second stuff that I was talking about, the evolutionary stuff. I'd like to thank especially also Eric Belfield, who's my laboratory manager and who has major inputs into all of these projects we just talked about. The SALT project is a collaboration with Andrew Smith in our department, with Richard, I've just mentioned, who's I established a collaboration with when I first arrived. And the evolutionary stuff is a collaboration with Lawrence Hurst at the University of Bath. Funding was from BBSRC and from Kaust King Abdullah University of Science and Technology in Saudi Arabia. I'm currently not funded at all, just so that if ever you come to review any of my grants, just think of me. And finally, I'd like to make a plug for our plant biology textbook that I and a number of John Innes and ex-John Innes people have authored. It's increasingly selling very well. We're not beating the major US textbook, Teitz and Ziegler, yet, but I'm very hopeful that we will. So please, Europeans, keep buying it. And finally, a plug for my own popular science book, for those of you who like reading those kinds of things. It had a very nice review from Jenny Uglo, which said, brilliant, a bravura performance, a privilege to watch a subtle and daring mind at work. So, so with whatever subtlety or daring I've got left at the end of all this, I'll be very happy to take any questions from you. And thanks for your attention.